But be sure to download the materials and check those all out. Now we are moving on to our first speaker, which is going to be Braden Elliot. And Braden, sorry, let me stop sharing. Yeah, so we're going to move on to our next speaker, Braden, who's going to introduce us to linear regression. Take it away.
Hi, my name is Braden Elliot. I'm an environmental scientist with the State Water Resources Control Board. Welcome to the regression session. I hope that you're enjoying your day so far and learning a lot. I'd like to talk to you about simple linear regression, which is a tool you can use to relate a continuous response to a continuous predictor. Let's locate ourselves on our map or our decision tree of what analysis is appropriate for the data I have in front of me. If your response is not categorical, your predictor is not categorical, your response is not a discrete integer, your relationship between the response and the main predictor is linear, your response is not binary or proportional, and your response is continuous, which is a long way of saying your predictor and your response are both continuous. Linear regression is the tool for you. A little bit of background about linear regression, or the simple linear model. It's an application of an equation you may remember from at some point in your life in school, y equals mx plus b. In this case, those letters stand for the response, y, being equal to some coefficient, m, times the value of a predictor, x, plus an intercept, or a grand mean. And statistics often interpret this slightly differently, but the form should still be familiar. If you just put the intercept before the coefficient times the predictor, and then because no model is ever 100% accurate, at least not in ecology, there is some residual variance between the predicted value of response and the actual value of the response, which we capture as residual variance of the letter sigma. So statistically, the simple linear model looks like y equals beta naught, this is at the bottom of the screen, plus beta 1 times x. If you had multiple predictors, you could list them here, like plus beta 2 times x2, plus beta 3 times x3. But in this case, we only have one predictor, so beta 1 times x, plus the residual variance, sigma. This model makes some assumptions that we need to ensure our data meet in order for the model to tell us something useful and representative. First, the model assumes that there is a linear relationship between the predictor on the x-axis, also known as the independent variable, and the response on the y-axis, also known as the dependent variable. The model also assumes that the predictor is independent of the response, that errors are normally distributed, and that those errors have even variance across each variable. That is to say, there is no heteroscedasticity. The model also assumes that the predictor is measured with no more error than the response. If you have crappy predictor data, it's going to be hard to get good response data or predict good response data. A linear relationship, just to remind you and give you a bit of a visual aid, you could think of as being able to draw a straight line through a cloud of points on an XY biplot in such a way that that line captures the relationship between those two variables. Here we have information about some people, some humans, in which height, the predictor, is on the x-axis here, measured in centimeters, and weight, the response, is on the y-axis here, measured in kilograms. And the relationship between those two looks linear, and the line captures that linear relationship. But who wants to hear about people? This is an ecology themed day, so let's talk about fish. Here's a rough example, the rough being this fish. Can we predict the weight of that fish from its length? It might be useful, not just in quantifying the relationship, but also in predicting. the harder to measure variable, weight, at least in a boat, it's hard to measure, from the easier to measure variable, length. If there is a relationship and we can quantify that relationship with a model, then we can predict weight from length and use the model to infer weight, which we couldn't measure in the future, from a length, which we might be able to measure in the future for a new sample of our population. So let's go to the code and work this example out. Here we are in RStudio. On the top left is the script I'll be following. Bottom left, the console where the pieces of the script that I run will go to R. And then in the bottom right, you'll see some pictures pop up when we draw plots. So let's do a little bit to prepare our workspace by loading a couple libraries. And then let's pull in some data from one of those libraries. Let's interrogate this data a little bit. What's the structure of data? As in, what are our variables? What kind of information is in them? Numerical, integer, factor, and so forth. What are some quantitative summaries of those data? And what are the first few lines of those data, just to get ourselves acquainted with it. Here we're looking at the variable length and the variable weight. We're going to relate those two together. So let's just first drop all lines of our data set which don't have both of those pieces of information together, because we'll need them both in order to relate them mathematically. So we'll take a subset where neither of those variables is a not available or NA value. And then let's plot those two variables against each other. Doesn't look very linear to me. It might be monotonic, but you can see that if you were to draw a line through it, a curved line would be a better fit than a straight line. And that the points are more tightly hewing to that line on the left and much more spread out around that line on the right. So the residuals would funnel in a sense, and we're looking at some potential heteroscedasticity. So we can transform our data in order to meet the assumptions of our model by log transforming both the length and the weight. Now, what does that look like? Much straighter line, much more linear relationship. And we'll drop some of those low values because the relationship falls apart at that point. Now let's move on to analyzing our data. Here I'm going to call a linear model in which we predict log of W, log of weight, by the log of length using our data set in question. We're going to call that model rough LW. And then let's look at the summary of that model. We can see the call, we can see the residuals, which we'll interrogate shortly. The coefficients, which are the nuts and bolts of the model itself. The estimate for the intercept is beta naught, or B, depending on which version of the equation from a few slides ago you're preferring to think of. And the estimate for log of length is the coefficient associated with our predictor variable. So if we were to write out the equation in the way a statistician might, we would say that y, or the response, or the weight, the log weight in this case, of a fish, is equal to, first, the intercept, negative approximately 10.8, plus the coefficient associated with length, which is approximately 2.9, times the log of the length value of that particular fish, plus whatever the difference is between that value and the actual value. But the predicted response, or you might see it written as Y with a hat on it sometimes, would be negative 10.8-ish times 2.9-ish, or sorry, plus 2.9-ish times the value of your predictor at that moment. And then at the bottom of the summary, we have our residual standard error, and then your R squared, which is important. It tells you how much of the variance in the data is explained by the model. And it's almost 1. It scales from 0 to 1. So 0.989 is a very large R squared value, especially in biology. And then our F statistic tells us about the model fit. It's a relatively large number on 1 in 678 degrees of freedom with a relatively low p-value. So you think the model is working pretty well. And let's just do a little bit of validation. Let's look at the residuals of that model. They seem to be approximately distributed in a bell-shaped pattern according to this histogram. But we can more precisely determine whether they're normally distributed by giving them a normal quantile plot treatment. The greater that line is, the more normally distributed they are. The line looks pretty straight to me for a normal QQ plot. And then let's just plot those residuals against the predictor. So the predictor log length is on the x-axis along the bottom, and the residuals are on the y-axis across the left. And it looks, as they say, like a starry night. There's not a clear pattern. For comparison, on line 47 here, let's run a linear model simply relating weight and length untransformed. So predicting weight by length, and take the residuals of that model and plot them against length. So the same plot that we're looking at in the bottom right now, but if we had not transformed the variables in the first place. Remember, we're now looking at a residuals plot, not the plot of the data itself. So we can see that there is a clear visual pattern. It does not look like a starry night unless you are a dinosaur at the end of the age of dinosaurs. And you can also see some what we call funneling, where the variance among residuals is less at low values of our predictor on the left, and it is greater at high values of our predictor on the right. So this also illustrates heteroscedasticity. That's why we transform the variables in the first place. Now, to present the model here to an audience, we can use numbers, and we should, but we can and also should use pictures, because people learn from pictures very well. So I have here a ggplot command to draw a relatively pretty picture in R, illustrating the data and the trend in those data. So I'll run the command, and then here is a plot of the relationship between the length and weight of these rough fish, at least the log length and the log weight. And since I have log transformed the values, I've chosen not to include units, but it's good practice to include units. So going back to our rough example, how would we write up that model? How would we share that model with our audience? A quick, simple presentation in the text. You might tell your audience that length significantly predicts weight among rough, and give them some quantitative measurements of that. The R squared is, the decimal should be 2 spots over. The R squared is round up to 0.99, which is about as close to 1 as you can get without being 1 to that level of detail. Your F value on 1 in 678 degrees of freedom is a relatively large 61,760, and your P value, ideally you'll report an exact P value, but in this case there were more than a dozen zeros between the decimal point and the non-zero digits, so I've simply left it as a P of less than 0.01. And then graphically, the relationship can be shown much the way I drew it up with that ggplot command back in R. So there's a simple linear model. I hope you feel empowered to go out and make sense of 2 numerical continuous variables which have a linear relationship using this tool moving forward. And I hope you are also now prepared to extend upon this linear model through the rest of the regression session. Please feel free to send any questions or comments to me at braden.elliot@waterboards.ca.gov. Thank you for your time and attention. Have a good day.
Thanks, Braden. There are no questions in the Q&A, so as a quick reminder, if you have a question or a comment you want to make, you can feel free to insert it into the Q&A, or you can use the raise hand function, and I'll do my best to figure out how to unmute you. Also, I would encourage you all to check out Rosie Hartman's Twitter account right now, because she's adding limericks for each presentation in our micro training session. Rosie, why don't you go ahead and throw your Twitter handle in the Q&A. We have about 5 minutes until, unless I'm mistaken, yep, we have about 5 minutes until our next presentation. So I will just hang tight, and if anybody has any questions or comments, feel free to raise your hand. Otherwise, we will just patiently wait, collect our thoughts until the next presentation. This may be oh, we do have a new question. It is from a name that I do not know. I believe it's multiple names smashed into one. Unless that is your name, in which case I apologize. This may be a silly question, but I'm new to R. Is the decision tree publicly available? Can you add to that? What do you mean by decision tree? Do you mean the R handout?
Jereme, we can ask to unmute her microphone if that would be easier rather than having her type it.
Oh my goodness, that's why you're running the show.
Okay, hi, can you guys hear me?
Yes.
Yes.
Okay, so I was referring to the decision tree that was used in the talk to determine which test to use depending on your data.
Yes, so that is going to be in a lot of the handouts, but since I recorded my talk live for the introduction, I wasn't entirely sure what I was going to cover. So I'm planning on uploading the recording of my talk itself to our website. And I also will include, I will create a handout in the next few days that has those documents. So those will be there. It just may take me 24 hours.
Oh, no worries. Thank you so much.
Thank you. We've got another couple minutes for questions or comments. If anybody else has any thoughts. One of the questions is, please remember to take note of your favorite presentations and favorite posters, and be sure to vote for those. Also, there's a question from Li Minghe who says there's no data set included for this talk. Unless I'm mistaken, sorry, Braden's in the field actually. He got called in the field, so I'm trying to field his questions. Unless I'm mistaken, that was a data set from the FSA package. And so anytime there's not a data set, if you just open the code and look at the first few lines of code, hopefully there's a link to the data set, or it opens a package that has that data set automatically loaded into it. I am not seeing any other questions. So we're just going to hang tight until 10:40. But feel free to chime in if anyone has a question or comment.
Jereme, while we're waiting, would you mind if I read a poem that was submitted by someone who wasn't available yesterday during the poetry slam?
I would, except we just got a last.
Oh, sure. Go ahead. Take that question. Yeah, go ahead.
So this is from Dennis. From interpreting the log transform rough data set, is it valid to say that the length is a predictor of weight, or is it more accurate that log length predicts log weight? My perspective is that both of those are correct. They are, when you're doing a transformation, you're essentially saying it's a predictor, but in this special way. And anytime you have a log-log transformation, that's called a power law. So I would say length is a predictor of weight, but the relationship is a power law relationship or a log-log relationship. With that, we are out of time. So we're going to move on to our next talk, which is going to be Tyler from FishBio, who earned his MS at Kansas State and his PhD at University of New Mexico. And Tyler's going to be talking to us about logistic regression.
Hello, everyone. Thank you for coming today. Today, I will be talking about logistic regression, a commonly used statistical analysis for certain types of ecological data. First, I would like to thank Jereme Gaeta for organizing this micro training session and doing all the legwork to post the code and supplementary material on GitHub. This is going to be a great resource for IEP. I'd also like to thank my co-author, Matt Peterson, for helping me dig through all the different IEP data sets to come up with an interesting case study, and also for double checking my code. So as ecologists, most of us know that not all the data we go out and collect in the field are continuous and normally distributed. A lot of times we're interested in binary responses. So we might be interested in counting the number of dead versus alive individuals. We might be interested in the number of occupied sites versus empty sites, or healthy versus diseased individuals, or even the number of females versus the number of males in the population. This binary data can also be converted to proportional responses, such as we might be interested in variation in mortality rates against some explanatory variable. We could be interested in the proportion of occupied sites and how that changes over time. We could be interested in comparing infection rates across different diseases, or even comparing sex ratios across different populations. So by now you've probably seen this slide. This is a great flow chart for thinking about how to run statistical analyses. And so when we're dealing with binary response or proportional data, this puts us in the realm of logistic regression down here. First, a little theory. With binomial or with binary data, we have to think about a different statistical distribution, and that is the binomial distribution. So if you're familiar with this, if you're not familiar with this, think about a sequence of independent trials with two possible outcomes. So again, you can flip a coin 10 times and count the number of times you get a heads. Each time you flip a coin, it is independent of the previous time, and there's only two possible outcomes. And P is then the probability of success, or of getting a heads. Therefore, 1 minus P is the probability of tails. So for any random variable X, we call it binomially distributed with N trials and P probability of success. Algebraically, we can calculate the probability of getting exactly K successes as P to the Kth power, and you can see how we can calculate the probability of the exact number of failures. We also can calculate an expected value, or mean, of this random variable X, which depends on the number of trials times the probability of success. And you can see here how the variance around this value is also depends on number of trials and the probability of success. Now, one of the challenges with dealing with this type of data and wanting to model it against certain explanatory variables that are continuous is you need to have a link function. So the link function provides this relationship between a linear predictor and a response distribution that ranges from 0 to 1, 0 or 1, or ranges from 0 to 1. For the binomial distribution, the most frequently used link function is the logic transformation. And this transforms your probability to a log odds scale. So instead of estimating the probability of an event happening, it's estimating the odds that an event will happen. And so here is the calculation to convert probability to log odds. And the important thing with this is the new range is now your probability now is from negative infinity to positive infinity. So as you can see with this table, I took this table from Analysis of Ecological Data by Zur et al. In 2007. And here on the top row, you can see any number of probabilities and then their conversion to the log odds. So you can see now your response variable can take on values from negative to positive. So moving on now to our case study, I thought it would be kind of fun to look at some data on Sacramento split tail, Pogonictes macrolepidotus. So I used pulled data from the Delta Juvenile Fish Monitoring Program, specifically the beach seine data, because it's a good record of juveniles and the number of juveniles produced each year. So some relevant information, biology of split tail. So spawning is triggered by day length, increased flows, and when water temperatures reach 14 to 19 degrees Celsius. And peak reproduction is typically from March to April, but this can extend on either sides of those months. So we might think then that when the juveniles start showing up in the beach seine data is when water temperatures start to warm up in the spring. So we ask the question then, does the probability of split tail occurring at a station vary with water temperature? And we can also ask the question, does the proportion of stations where split tail are present vary with water temperature? So moving on now to the analysis. So we've pulled together this data set and we filtered it down and did some data wrangling so that we're only dealing with data from 2001 to 2010 and only the catch of split tail at different stations in each month. So here you can see the data in R where we have the year, month, station number of split tail captured, and the water temperature that was measured. And again, I should point out that this RDS file is going to be available on the GitHub site. So our code then filters this data to only include the months February through June, so that late winter, spring period, and this makes for a cleaner analysis. So our first question, does the probability of split tail occurring at a station vary with water temperature? So here we're going to use presence-absence data. The code provided in the supplementary document shows how to convert catch data to presence-absence and shows how to plot it. And this is what it looks like with presence-absence on the y-axis and water temperature on the x-axis. Now with this data, it's really hard to see if there's any patterns at all. So we'll go ahead and run this model. For those of you who are familiar with R, you may have seen this GLM function before for generalized linear modeling. We use that function to perform our analysis. PA here is our response variable. It's in our data set. Water temperature is our explanatory variable in our data set. And the most important thing here is to set this family argument to binomial, because this tells the GLM function that we're dealing with binomially distributed error, as well as setting the logit link function. So we can look at the results of this model, and we can see that water temperature is significantly positively associated with the probability of split tail showing up in the beach seine data. Well, that's not the whole story. You also have to ask, how well does this model fit our data? So many of you may know that R has a plotting method for the model objects that are created that will produce these diagnostic plots. However, when dealing with presence-absence data, it's really hard to interpret or make any sense out of these diagnostic plots because typically, for this one, you'd want to see almost no relation, no patterning, and this one you want to see them fall along the lines. But it's hard to tell with this. So another option then, and I provided the code to perform this other assessment in the supplementary document, is to break the data up into subsets of different water temperature ranges, and then to calculate the empirical probability of of split tail being at a site and see how well that compares to our fitted relationship. So this plot here has the probability of split tail being present at a station on the y-axis and water temperature again on the x-axis. Our lines here are our predicted relationship with 95% confidence intervals. And then this method where I split the data into 5 different subsets and to compare. So we want these points to fit very close to this line. And for some parts of this data set, it works pretty well. But you can see out here at the end at higher temperatures, our model really does not do a good job. All right, so now we'll ask the question, does the proportion of stations where split tail are present vary with water temperature? So the code here basically takes that presence absence data and converts it to the proportion of stations sampled each month and that have split tail present in them. And then it also calculates a mean water temperature across those stations. And we can plot that, and we can see that there does appear to be some influence of water temperature on the proportion of stations with split tail in them. So we can formally test that again using the GLM function in R. And there are 2 different ways of dealing with this data, setting up this model. So one, we can use the actual proportions that we calculated as the response variable. You can see mean water temperature now as our explanatory variable. Again, we set the family to binomial. But if you're using proportions, you need to tell the GLM function how many stations were sampled to get the observed proportion of sites occupied. So we have to use this weights argument here, where n is the number of stations sampled. Alternatively, we can use a binary response variable where we have the number of sites where split tail are present and the number of sites where split tail are absent. So that's our response variable. This is our explanatory variable. And you see, when we do that, we don't have to use the weights argument. So we'll take a look at the results from this model. Again, it's pretty clear that water temperature has a positive effect on the proportion of sites where split tail are present. One more thing to point out with this data and these results here are looking at residual deviance. And when you set the model up this way, you set the data up this way, you have to check for over dispersion. So here we have our residual deviance is much greater than our degrees of freedom. So this is an indication that there is over dispersion in this data. But don't worry, we can easily fix that by using the quasi-binomial family. So you just change this family argument here from binomial to quasi-binomial, and then you'll get a more appropriate error structure in your results. So we can plot the, so when we plot the predicted relationship, which is the black line and 95% confidence intervals, the gray lines, we could see that over the top of our observed data, it seems to look like there is a pretty strong relationship there. But we also, again, we have to check how well that model fits the data. So now we can use those typical diagnostic plots to assess our model fit. Unfortunately, we see a little bit more patterning here in this residuals versus predicted values. And our normal QQ plot, these points don't really fall as well on the line as we would hope. But that's okay. We're only using one explanatory variable, and we could probably get a better fit if we increase the number of explanatory variables in our model. So with that, the important things to consider with when reporting on the results of a logistic regression, you definitely want to report the model coefficients and their standard errors. You may want to report p-values, but some people these days are on the fence with p-values, and especially with if you end up using the quasi-binomial family, those p-values are only going to be approximate. So you need to be very careful if those p-values are close to your 0.05 cutoff value. You can always include additional predictor variables to improve the fit of your model. Just like multiple linear regression, you can have multiple explanatory variables in a logistic regression. You can also set up different models to compare how well different models fit using multi-model inference and certain information criteria. And then lastly, with this particular analysis and these questions, we're not accounting for imperfect detection. So there could be multiple reasons why split tail is present at a site, but it wasn't captured in the beach seine. So in that case here, any inference that we make with this could be biased because we're not accounting for detection. So with that, thank you. I'm open for questions.
Thank you very much, Tyler. So far, we have no hands raised. I'm not seeing anything in the chat or in the Q&A. So if anybody, oh, here goes one. So this is from G&G. Do you have any advice if you believe your data relationship asymptotes at a probability or proportion lower than 1?
I would say try to find a different test because, I mean, with logistic regression, one of the challenges is when you get really, really high probabilities or really, really low probabilities, you're running into an issue that's called testing on the boundaries. And these tests kind of fall apart at those levels. So, you know, I'm really not sure what you would do if your probabilities are really, really close to 1 or really, really close to 0. That might be an indication also that you might need a more advanced test like the Zero-inflated, because your data, one of the reasons that you might have those values really, really low or really, really high might be you just have too many zeros in that data set. So hopefully that answers. This is hard.
Yeah, so to answer that, I think that's case specific, in that if you have a situation where, you know, your values start low and or high, but if they start low and you see them go up and they just stop short of 1, then logistic regression is going to be great. But if they start at 0 and then go up to 0.001 and asymptote there, that's when you really have that problem and you need to look a little deeper. Let's see. I'm not seeing any other hands or any other questions. I would also encourage folks that if you have a question that you post and we don't interpret it the way you mean it, feel free to elaborate on that question in the chat, and we can either circle back to it now or during the open Q&A. Another one from Gonzalo. How do you decide whether to use zero-inflated versus binomial modeling? Well, I'll field that one. And the answer is, I'm not going to tell you, because in approximately 1.5 minutes, Rosie is going to give us the first of 2 talks that tell you exactly how to do those things. So thank you for the segue, Gonzalo. Any other questions before we move forward? All right, well, thank you very much, Tyler. Our next speaker, as I just mentioned, is going to be a pair of talks by Rosie Hartman, who earned her PhD in ecology at UC Davis. And this is going to be a set of talks. Unless I'm mistaken, we're going to have a bit of a break in the middle so we can have Q&A on the first session, or the first section, and then we will move on to the second section of the talk. And again, her first talk is going to be on regression with negative binomial distributions. So with that, thank you everybody for sticking around. I encourage you to continue to add questions, and Rosie, go ahead and take away. I'm not getting any audio, Christine.
Hang on 1 second. I'm having technical difficulty with her file. Okay, let's try this again.
Hi, folks. Welcome to our next session of this fantastic IEP workshop training. We're going to
Christine, we
continue
have
on
audio,
the
but
theme
no
of
visual now.
regression analysis. This time talking about, instead of measured data, count data.
So you're not able to see it?
We just saw your desktop, or at least I did. I'm assuming everybody's seeing the same as me.
Okay. I'm sorry. Let's try this for the 3rd time. I think this is gonna work.
It is a charm, as they say.
Okay, hang on. Here we go.
Hi, folks. Welcome to our next session of this fantastic IEP workshop training. We're going to continue on the theme of regression analysis. This time talking about, instead of measured data, count data. So where are we on our little flowchart of different potential analyses? Well, we are starting over here. Is our response categorical? No, we're talking about count data right now. So not lengths of fish or types of fish, but numbers of fish. Predictor's categorical? No, not right now. Response catch data? Yes, so we're in discrete integers. Is our response zero-inflated? We'll talk about zero-inflated next. For now, we're gonna go no. So we're gonna deal with negative binomial or Poisson regressions. And I'm gonna give you a little spoiler here. You almost never use Poisson regressions. Data almost never fits. You usually have to go to a negative binomial or similar. But we'll talk more about that in a sec. So, assumptions of a linear model is that, as we discussed this morning, the true relationship is linear, errors are normally distributed, there's equal variance around the line, and independence of observations. But fish are not normally distributed. They're never normal, they're weird. How do I run a linear model on fish data? So normal distribution, as I'm sure you all know, some randomly generated data, nice little bell curve. I took the striped bass data from the fall in the water trawl, calculated the total number of striped bass per monthly survey, and this is what the histogram looks like. That's not even close to normal. So what do we do? Well, one thing you can do is use data transformations, which many of you have probably heard about before. One of the most common is taking a natural log or log base 10 and applying that to all your data. If you have zeros in there, you also have to add 1 before you log transform it, because you can't take the log of 0. And this kind of feels like cheating, but it's totally legit, so long as you do the same transformation to all your data. However, it can make interpreting the results a little difficult because the coefficients can be on a log scale, and it can introduce some biases in your data. It works just fine if the dispersion is low, so up around here, and if the true mean is relatively high, then it doesn't really matter. You'll do just fine. But if you have a lot of dispersion like this, or a relatively low mean, sometimes you can get errors when you log transform your data, especially if you try and use it for making predictions. So it could be just fine for your data, but be careful and make sure you know what you're doing. In the materials that are online with this presentation, there's some links to some articles that kind of go through the issues involved. So just kind of know that there are issues involved for now. Okay, so if we're not going to log transform it, what do we do? Well, it's always better to not mess with your data and to actually run the statistics on the data rather than the transformation. So this is where generalized linear models come in. They use all the same techniques as the classic linear models. They just assume a different underlying distribution. The Poisson distribution is one that you probably remember in Stats 101. You learn about it right after the normal distribution. It assumes that some events occur at a constant rate. So the number of events that occur in a given period of time is going to follow a regular Poisson distribution, where the mean is equal to the variance. Poisson is also French for fish, so it should work for fish, right? But the mean is never equal to the variance, because a Poisson distribution assumes that you're trawling along, you encounter fish at a pretty constant rate, so the number of fish in your trawl is going to be pretty constant. You know, fish don't act like that. We all know that. When your data are over dispersed, the mean does not equal the variance. For example, in our striped bass data, mean is 138, variance is over 1 million. This always happens because they're trawling along, and you'll end up with a school of fish. You'll end up with a dry spell with no fish. Fish will hide in the weeds. A sea lion will scare some fish into your net. Like, it never works. So what do we do? There's a couple of tricks that we can use. The first is a quasi-Poisson distribution that includes an over-dispersion parameter. I don't use this one that often. I don't know a lot of people who use it super often, but it's an option. Feel free to explore. The one that's used a little more often is negative binomial distribution, which models a number of successes in a sequence of independent and identically distributed Bernoulli trials. Yeah. The important thing is it means that the mean doesn't have to equal your variance, and it's related to your mean based on some over dispersion parameter. Fish don't quite do this either, but it's better. Let's go to R real quick. Come on. And we can actually do a demo. So we're going to load some packages here. Tidyverse and Liberdate are just ones to make the data manipulation easier. Mass we'll need for the negative binomial distribution, and these others will be to help us interpret the results. So starting with data from the Saltwater Trawl, I already downloaded this from the CDFW ITP site. I found the Catch Matrix updated tidy version. I'm so excited they have a tidy version now. I'll read that in. I also have a CSV of water year type assignments. So dry, above normal, below normal, etc. I'm going to use that to help model our data. We're going to join them together. And I'm going to run through this data manipulation really fast. If you have questions, feel free to reach out later, but I want to jump right to the statistics. The subset striped bass and calculate the total number of striped bass caught per monthly survey. Now, whenever I get a data set, first thing I do before I run anything is just do a quick plot or two. So, looking at catch over time, color coding by year type. Okay, we have some really high catches in the beginning, then it got a lot lower. Recently, it's been really low. See what a histogram looks like. So yeah, we have a lot of really small values, very few big values. Now, can we log transform it? Yes, we actually have a pretty nice bell curve when we log transform this data. Note we had a little error message, remove 2 rows containing non-finite values. So we probably had some zeros in there. We should have added 1 before we log transformed it. So now we're going to do a linear model just from the log transform catch data. To do this, we use the LM command for linear model. The response variable is going to be the log of catch plus 1 because we had some zeros in there. And the predictor variables are going to be year and water year type. Just run that. Look at the summary. Okay, so we've got what we put in, residuals, and then our coefficients, which is really what we're interested in. We see that there was a significant effect of year, a negative effect, so higher in the beginning, lower later. And wet years are significantly higher than the intercept, which was the above normal here because it's first in the alphabet. Got a decent R squared, 0.61, okay? So after we run a model like this, we have some significant p-values, but we always want to check our assumptions and look at our partial residual plots. So we can use the plot command to look at these diagnostic plots. And in the online material, there's some good explanations of everything you're looking for in these diagnostic plots. Residuals versus fitted, this is kind of look like a cloud and pretty much does. With normal Q2 plots, all the points line up right on that 1-1 line, and it's a little bit iffy here, but it's not terrible. Scale location, this also kind of looks like a cloud, which is good. Same with residuals versus leverage, this is where you check for outliers. And all the points need to be inside the dotted line. We don't even see the dotted line, so we're good there. But Q-plot wasn't great, so I feel like we should probably try and use a different distribution. We're gonna try a Poisson distribution. Like I said, this probably won't work, but we're gonna give it a go. Woohoo! So the GLM stands for generalized linear model, and it's run it the same way as our linear model, but use family equals Poisson. And when we ran the summary on this, all of these were significant. They're great. Woohoo! Poissons always do this. They give you highly significant p-values on everything, but we check our diagnostic plots. They're looking a little iffy. So this one, which looks like a cloud, instead we have a trend where there's greater dispersion off the line for higher values. That's the bad side. Normal QQ plot, I got a lot of points way off the line. Location, we have sort of an increase in that red line. Not good. Residuals versus leverage is really looking bad. All sorts of points outside the dotted lines. You can also use the simulate residuals function from the Dharma package to give you a different set of diagnostic plots I really like, because they print the results of the test right on there, and when there's a problem, it's in red. So it says problem, problem, problem. Very easy to interpret. So over dispersion, we know it's one of the problems that we could have. So if we do this test for dispersion, we see the p-value is 0. This is our simulated value. These are all of our actual values carried over dispersed. So now let's do a negative binomial distribution. We'll use the GLM-NB function. So we don't have to specify the family because it's in the function itself. And same formula, catch by year and year type. And here we don't have all those highly significant p-values, but it looks pretty similar to our log transform version. We have a significant effect of below normal years being a little lower. Year type, or sorry, year is really significant. Wet years are still significant. So kind of passes the laugh test. Now let's look at those residual plots. We can't use just a plot command on the negative binomial. It doesn't come out right. But we can use this, and we see that all of those tests that were in red before are now in black. And I say deviation non-significant. No significant problems detected. Look at me, no problems. If we choose test dispersion, now we see that the red line is kind of in the midst of all of our actual data. So we've got a good model. It's decent. Now we need to make sure we can interpret this model. So we can look at the effects, the means of the different categories of our water year types using the NM means function. And this tells you the mean for each of these years, standard error, confidence intervals on the log scale or on the response scale, because the negative binomial uses a log link. I should say that earlier. So that's what your actual number of fish will be in this response column here for each of these water year types. Those are the means. Now, the best part, though, is the pairwise comparisons. So we can look at above normal versus below normal, above normal versus critical, etc. We don't get from just our summary of the model. And it'll give us the p-value of which ones are significantly different. VisReg is one of my favorite packages for visually looking at this. We can see the effect of year when water year type is held constant, and the effect of water year type when year is held constant. This is again on the log scale. If we want it on the response scale, you can just put scale equals response, and now that looks almost exactly like our data. Like, hey, that's probably a pretty good model if it looks like the curve in our data. There you go. So I don't think we're going to have time to cover offsets, but very quickly, if you want to model catch per unit effort instead of catch, negative binomial and Poisson will only accept count data. They won't accept fractions. So you'll have to use a offset, so I'll model catch and then have offset log of volume rather than modeling catch divided by volume as our predictor variable. And I'll leave it at that. I would absolutely love to take follow-up questions, meet with you one-on-one, whatever you like. And have an absolutely lovely rest of your day. In a few minutes, I'm going to be back to talk about data with lots of zeros.
All right, thanks, Rosie. So we have about 4 minutes. I may want to start Rosie's next talk a little bit earlier just because it's slightly longer, but we have plenty of time for questions. Right now I'm seeing no raised hands and no questions in the Q&A. In the meantime, I'd just like to note that most, or at least a lot of these handouts cover a lot more material. For instance, the offset term that Rosie was just talking about. Another example, I can definitely speak to mine. In my next talk, I think in my talk I make it through page 20 of the handout, but the handout is a 37-page handout. So I definitely encourage you all to go ahead and look at those handouts, if not actually read them top to bottom. Still not seeing any hands or any questions, but we'll just hang tight. Well, actually, I'll go ahead and ask you a question, Rosie. So the offset, can you elaborate that on about that just a little bit? My understanding is the offset is kind of like a effort term. Is that,
Exactly.
What are your thoughts on it?
Yeah, so in a negative binomial model, it's expecting count data. So it needs integers. If you give it decimals, it goes, that's not count data, those are decimals. You can't have half a fish, that doesn't make sense. But we usually want to standardize by effort. So instead of having, your response variable be catch per unit effort, you put effort as a term in your model. And it'll usually be because a negative binomial or Poisson model uses a log link. So it, without going too much into the theory there, you want to use the offset of the log of your volume if your effort is in volume or if it's in time or something like that. And that allows you to standardize for the fact that, a catch of 5 fish, it could have been from a really short trawl, or it could have been the fish were really scarce in a really long trawl. So that's kind of how that works. And I do go over that in the handout that's provided on the website.
Perfect. Yeah, I had another question, but we have questions in the chat. So Christina Bergy wants to know, why go for a negative binomial with catch data at all when you're going to have a tons of zeros usually? I'll let you go ahead and answer that.
Well, that's the next talk is on zero inflation. It turns out that most of the time, a negative binomial actually does a really good job at modeling data, even when it has a lot of zeros, and you think you might need some sort of zero-inflated model. Often the normal negative binomial actually fits pretty well. If you have a lot of zeros and you think that there's a different cause driving the zeros from the counts, then you want to go to something that's zero-inflated, or if you can't get the normal negative binomial to fit.
Yeah, so a great example of that Rosie discussed with a group of us is that if, let's say, salinity may be driving whether or not you're getting that zero, but something like temperature is driving the actual number of fish you're seeing. One last question that I'll just hit really quick. Can you include random effects? The answer is yes, and stick around for session 8 the next session after lunch, where I'm going to talk about random effects specifically. And with that, we have to move on to Rosie's next talk, which is, as she alluded to, going to be that zero-inflated discussion. So Rosie/Christine, take it away.
All right, everyone. Welcome to the second part of our fun day with count data. As the fantastic training in our statistics, etc., continues. Now, if we go to our flow chart, our last presentation talked about negative binomial and Poisson regressions. We started with our response not being categorical, our predictors not being categorical, and looking at data that are discrete integers. Now, the last presentation, we said the response was not zero-inflated, so we went to negative binomial or Poisson regressions. Well, now we're going to see what happens when your data are zero-inflated, so you have too many zeros. And we'll look at zero-inflated negative binomial models, as well as hurdle models. And we're going to talk about how sometimes you don't even need a special zero-inflated model. You can just use a negative binomial. But I think we can all agree that the regular old linear model, with its assumptions of the true relationship being linear, errors being normally distributed, homoscedasticity of errors, and independence of observations, is not going to really work out when we start getting these complicated count data sets. Because delta smelts, you can't catch them. They school, so you'll end up with more of them than you would expect in some trawls. They're really easy to misidentify as Wakasagi or longfin smelt or whatever. And yeah, they just do not follow a normal distribution. So how do we run a linear model on smelt data? The last presentation, we looked at the striped bass data, which had a lot of zeros. This histogram of the smelt catch is even less normal looking than the striped bass one, though. We have even more zeros. So the first thing that we had talked about with in the previous lesson was the first thing you could try is try log transforming your data. This can often turn it into a normal distribution that you can run a linear model on, but not here. This is the log transform version. We still have way too many zeros to fit a linear model to this. So the next steps that we had talked about was a Poisson distribution, but that doesn't even usually work if you don't have too many zeros. So what do we do about when we have this many zeros? We can try a negative binomial, but it's important to think a little bit about what those zeros mean, because you have different kinds of zeros. There's what are known as structural zeros. So we're not catching delta smelt because the habitat is just not going to ever give you delta smelt, or you're on land, or just areas where you're never going to see smelt, no matter how good things are. Too hot, too clear, too salty, etc. Then you have design or sampling error. This is a thing that we have to deal with in almost any data set, such as the fall midwater trawl is a midwater trawl and delta smelt tend to hang out near the surface. Or they're small and can escape the net. We have observer error. So you might catch a smelt, but think it's a Wakasagi, and so throw it back. I know I would. I'm terrible at telling them apart. Or you have smelt error, where the habitat was great. You should have caught a smelt, but there are no smelt. And so a regular old model is pretty good at figuring out some of these types of zeros, but not always all of them. So what do we do? Well, one technique you can use is called a hurdle model. Here, we take all our zeros and just do a regular old binomial model or logistic regression on presence absence. Then you take all the situations where smelt were present and look at what happens when what controls how many smelt you have when you have smelt. This basically treats all the zeros the same though, so it doesn't have quite as much information as some other types of models, but it can be useful in certain circumstances, especially when you have a strong feeling that there are certain factors that describe presence absence that are different than the factors that describe number of fish when they are present. Now, a zero-inflated model or mixture model treats some of your zeros as, as if we're doing a regular old regression where occasionally you have zeros for whatever reason. That's separate from the zeros that are due to, just being in totally the wrong place or something like that. So this gives you a little more information about what is causing those zeros. Also, it's important to know that often the way negative binomial models work, they'll deal with a lot of zeros. So you might not actually need to use one of these more complicated zero-inflated models if a negative binomial model will work. Times when you want to go to those zero-inflated models is when you really have a strong feeling there's different predictors that are causing the zeros versus the non-zeros. So let's go to R and do a little demonstration. So if you're following along with the online materials, this is the second part of the zero-inflated data count data code. But this time, instead of taking the formula from the data set and subsetting the striped bass, we're going to subset delta smelt. We're going to filter it down to just the delta smelt that are caught in Montezuma Slough from 1990 to 2020. I'm going to do some quick plots of the data, see what it looks like. Okay, so more smelt in the 90s than recently. Looks like maybe more smelt in dry years and above normal years. A little hard to tell. Now we do a histogram. And wow, we have a lot of zeros. Log transform that histogram, and we're adding 1 to the catch, because if you just try and log transform catch, the log of 0 is infinity, negative infinity, won't work. So we add 1 to each value, then log transform it, and we still do not have anything normal. So we're not going to even try running a regular linear model. We also discussed earlier that Poisson distributions almost never work. That's a demo of what would happen if we did it in the online materials, but just going to go right to the negative binomial model. The other thing that can happen when we start getting these more complicated models is that if your predictor variables are on very different scales, it can make the model really hard to fit. So we want to see if delta smelt are predicted by conductivity, temperature, and Secchi depth and year. But we have Secchi depth, which is, usually in the 1 to 200 range, year, which is 1990 to 2020, and conductivity, which is, from 100 to 20,000. And those are very different scales. The model's going to have trouble. What we're doing is scaling them. So we are dividing by the mean. We're subtracting the mean and dividing by the standard deviation to put them on the same scale. So now we have our negative binomial distribution. We're using a slightly more complicated model than the last one because we're looking at the catch at each station. And the stations are not going to be independent. So we're going to add a random factor of station. How to do these random variables and mixed models are going to be covered more this afternoon, but just know that is there to make sure that things stay independent. We're using a GLMM TMB function, and we're modeling catch as a function of conductivity, Secchi depth, year, and a random effective station. Family is n binom 1, which is negative binomial, and we've got our data. So what does that look like? It's a little more complicated, so it takes a couple more extra seconds to go through. Well, we see we had a effect of connectivity, psyche, and a highly significant effect of year. So that's, looking pretty good. Look at our diagnostic plots. Okay, so this is actually not bad. Our QQ plot is almost perfect. All the deviation tests that get printed on there are black, meaning you're in the clear, non-significant. However, going over here, you have quantile deviations detected. So there's some issues, and the combined adjusted quantile test is significant. So we've got some trends in the residuals versus predicted values that aren't really great for our models. It's not a perfect fit. Let's see, is it over dispersed? We can test our dispersion. So the p-value is 0.26. It's not over dispersed. It's, the red line is smack dab in the middle, but it's close. Is it zero-inflated? We knew we had a lot of zeros. Surprisingly, this is not zero-inflated at all. The red line is right in the middle of your predicted values. The model predicted just as many zeros as we saw, but we still have those quantile deviations. So what should we do about it? This is where we have to think, like, okay, we know there are a lot of zeros. Are there factors that might be causing the zeros that are different than the factors causing the counts? So with delta smelt, we know that there's been a long-term decline. We also know that they tend to follow psyche, connectivity, and temperature, but we've seen this decline that isn't really tied to changes in psyche, connectivity, and temperature. So maybe those extra zeros are best modeled by the year of collection, rather than psyche, connectivity, and temperature. So to do a zero-inflated model, we use the same GLM-MPMB function. We have first the formula for our count model, catch as predicted by psyche, connectivity, temperature, and station. We have VI, which is the zero inflation model. And we're going to just use year as the predictor variable for the zero inflated model. Family is negative binomial. Control optimizer stuff because it was having trouble fitting earlier, and so I Googled potential solutions, and this was one of them, and it works now. Don't worry about it too much, but know that when you get to these more complicated models, there could be some little tweaks you need to do in order to get them to run nicely. And you'll see that while a linear model, looking at the same thing, runs almost instantly, this took a couple seconds for it to think about it. What do we get? Okay, so this looks similar, but now we have two sets of coefficients. We have our conditional model coefficients, which is the counts, and then our zero-inflated model coefficients, which is the zeros. The conditional model, this looks just like a regular negative binomial model. Psyche, connectivity, temperature, p-values. This, the direction indicates the direction that the smell population would go. The zero-inflation model is a little hard to interpret the coefficients if you're not used to it. The estimate here is actually the number of extra zeros, or the effect on extra zeros. So a higher estimate here means more extra zeros. In the count data, a higher estimate would mean more catch. Here, it's less catch, more zeros. Okay, let's look at our diagnostic plots. All right, no significant problems detected. That's what I'd like to see. So even though the zero inflation test on the negative binomial model didn't show a problem, adding a zero inflated term fixed all of our problems in our model residuals. If we use our dispersion test and zero inflation test now, everything is in the clear and we're good. So, as I said, frequently you can just run that negative binomial model and it'll be fine, even if you have a lot of zeros. So don't jump right to the zero inflated model unless you really have a prediction for why zeros would be predicted separately from the counts. If you want to run a hurdle model, it's very similar. we can use almost the exact same syntax, except instead of family n by num1, we use family truncated n by num1, which means that we've cut off those counts at 1, and we're not going to have any zeros in our count model.
Sorry, folks, we're going to try and clear this up a little bit.
Need to think about it. To think even longer than the negative or the zero-inflated one. Looking at our summary, when we don't have any zeros in our count model, none of our predictors are significant, which is interesting. But we do have significant effects on the zero inflation model. Come on. So still looking pretty good. But it's just a matter of, you know, which model you want to use. You just have to think about what you know about the system, what you know about delta smelt. This model building is a bit of an art, and there's no like one true answer. So experiment with these different types of models, figure out which ones you really understand fully, which ones are the best fit for your data, and which ones do you have a conceptual model that makes sense biologically. And that's all I have. Thank you very much for coming. I'd be thrilled to take any questions either now or follow up with me afterward. My contact is on the online materials, and have a lovely day.
Thank you, Rosie. Sorry about those technical difficulties, everybody. I think we're going to try to make some adjustments before the start of the next talk. In the meantime, if there's any questions or comments, we have about 3 minutes, 2 to 3 minutes. There seems to be a new message. Oh, that's just information on how to submit. I do think Rosie's last statement is incredibly important in that as you move on to more advanced analyses, the path becomes less of a straight line. And it does certainly become more of an art. And one of the tools that I'd recommend you use is a log or a journal. When you're running analyses, I've literally done analyses that take months from the first time I run the analysis, or years actually, first time I run the analysis to my final product. And so I highly recommend having some sort of journal or log along the way that tells you, or in which you remind yourself why you made the decisions you made. Rosie, do you have any thoughts on that?
Yeah, just backing that up in terms of, this seems to be the stumbling block for most people as they're getting more adept at statistics. They'll pick up how to actually run the code pretty quickly, but then they have to decide which version to use. And that can be really difficult because there's this idea that, oh, there's a right way to do it and a wrong way to do it. The right way to do it is be clear about why you made your decisions. As Jereme said, write it down, have some backup for it, and be prepared to defend it. And there will be a million different ways of doing things. So just have confidence in yourself that you're doing things one version of right. Also, workshop with other people. You know,
Absolutely.
Make your decision and say, hey, does this decision make sense? Do you agree? Would you do it this way? Just to get some different perspectives.
Yeah, I think something that you know, our program coordinator Stephanie always says that's so important about IEP is that remember the community feel. We are a community, and the folks that are giving talks today, the folks that are much smarter than the folks giving talks today, all have answers, or at least can point you in a direction. So, you know, I can only speak to myself, and I'm pretty sure Rosie, but we do enjoy getting emails from folks. You know, when I'm beating my head against an analysis, sometimes it's absolutely great just to get a random email of someone saying, hey, I'm getting this error, or hey, I don't understand this this output. Can you help me out? The community is great. We do have a couple comments really quick. One from Dan that is absolutely spot on. I suggest just putting those notes straight into your comments. By comments, I'm assuming he means in your actual script. That's a great way to do it. I like to keep a journal on top of that because very often I will have 30 scripts from the first iteration of the model to the last iteration of the model. Before we move on, just really quickly, there's a question on interpreting zero-inflated model outputs. We're going to put a pin in that one and circle back to it in the open Q&A. But for now, thank you so much, Rosie. If there's more questions, again, please bring them to the Q&A at 2 p.m. We're going to move on to the last talk of this session, which is myself. And I'm going to talk about generalized additive models. A quick note, I always call them general additive models. So forgive the typo and the brain fart. Hopefully you learned something. Hi, my name is Jereme Gaeta. I'm a senior environmental scientist with IEP and CDFW. And today I'm going to talk to you about general additive models, also known as GAMs, which is a form of nonlinear modeling. Now, before we dive in, I'm only going to be able to cover about 1/3 or 1/4 of what I cover in the actual handout. So before you dive into general additive modeling, I highly, highly recommend you walk through the entire handout. Now, general additive modeling is most suitable when your response and predictor variable are not linear, not linearizable, or not sigmoidal. In other words, general additive modeling is really suitable when you have a curve or a wiggle in your data. Now let's start with a basic regression. I often find that the best way to understand an analysis is to understand the model notation. Now we're all familiar with this equation from algebra class, but that doesn't quite cover all of what a linear regression does. And that's where this equation comes in. Now it may look a little scary, but we're going to walk through it quickly. From our algebra equation, we know that B is the model intercept, which is the point at which X crosses 0. And in this case, it's actually 50. For this more complex, intimidating looking equation, that's just the alpha parameter. So it's the exact same thing. So I bet you can guess what M is in this other equation. M is our rise over run, and that is beta, where in our case, our rise over run is -3. So if we plug these values in, this is what our equation would look like. There's this other part, this epsilon, and this epsilon defines your variance or your uncertainty around this mean model prediction or the regression line. All it means is that our uncertainty or the observations vary around this regression line, and the structure of their variance is a normal distribution, which is shown here in this plot, where mu equals mean. The mean is 0 because the variance varies around that regression line. So the only term that we need to know is this sigma squared, and all that is the variance. Or in other words, if you were to turn this equation on the side and draw a normal distribution around the regression line, where the middle of that normal distribution is at the regression line or at 0, the variability or the width of those normal distributions would be your sigma squared, which in our case is 5. That works great when we have a straight line, but what does this equation look like when we have a curve in our data? Well, this is where it gets a little bit more complex. We still have an alpha term, an intercept term. We still have our epsilon term, and our error structure is the same. But now we have this f of x term, and all that means is that we are going to use some sort of unknown, undefined smoother function. The data set we're going to analyze is the YOLO bypass total aquatic macroinvertebrate data, and this is from their drift samples. In this plot here, I've natural log transformed the catch per unit effort data, and we are looking at it across the day of water year, where 0 is October 1 and 365 is the end of September. So what does this function do? Well, the next series of plots is going to show you kind of under the hood And again, I encourage you to read the handout for more information on this. So what it essentially does is it takes our data set and it breaks it into chunks. And at each chunk, it's going to build some sort of a model. In this case, we're using a linear model or a first order polynomial. The smoother functions can take a number of different shapes. However, what you notice is two things. The first is that these lines don't meet up. And what the model does to make those meet up is it takes first and second order derivatives. The other thing you'll notice is that at the data extremes, we have less data. That window width shrinks. And so what that does is that increases our uncertainty at the edges of our model. And you'll see that as we move forward. Now let's actually dig into the code. The model is surprisingly simple to code. I recommend you use the GAM function from the MGCV package. That's my preferred package. In this code, just like a linear regression, we put our response variable and we say it's some function, which is what the tilde means, of a predictor variable. However, we have this new term. That just tells it this isn't a linear relationship between catch per unit effort and day of water year or water doi. It's actually some smoother function. And this BS argument is how you define the type of smoother function you're going to use. In our case, and I recommend you start with a cubic regression spline. That said, I highly recommend you look at the help menu for this BS argument because there's a number of handy functions that you can actually use, different kinds of smoothers that are suitable for different kinds of data. And then finally, we just have defined the data set where our data comes from. Now, whenever I run a model, the first thing that I do, that I recommend all of you do, is hit the summary function. Right away, it's always good to actually check the formula that you are looking at in your summary. And in this case, that's what we are hoping to model, so that's great. And what I hope you see here, we only have one coefficient. If this was a linear regression, we would have an intercept and a slope coefficient, or an alpha and that beta. But here we just have the intercept. Another thing that's important to note is that this intercept isn't the same as a linear regression. This intercept isn't where the line crosses an x of 0. It's actually just the mean of our response variable. Another important term to know is this EDF. EDF stands for effective degrees of freedom. And really all that means is how wiggly is our model. So if our EDF is a 1, that means our model is a just flat straight line. And anything above 10 is incredibly wiggly, or in other words, there's going to be a lot of ups and downs. And then the final term that's really important to note is this scale estimate. Scale estimate is GAM speak for residual variance. We can plug in the intercept, we can plug in our uncertainty, but we have no idea what's going on with the smoother function. It's kind of a black hole. So we can't, like in a linear regression, report the coefficients. Somebody can't read our report or paper and then build that model themselves. They would have to have our data and our code to replicate this model. So that's an important limitation of GAM that you should consider before you take this approach. Let's start by evaluating the model fit. This is always a safe move before you spend a lot of time digging in your model itself. One of the first things you want to look at is your observed versus predicted, where here we have our observed data on the Y and our predicted data on the X. And really what you just want to see is to make sure that where you are estimating is about where the observations were. The more appropriate look at our observed versus fitted is in residuals, and residuals are observed minus predicted. Here we see that this fits as far as the eyeball test, a very normal distribution, which means our model's doing what it's supposed to do. Here we're looking at the same residuals across our predictor variable, day of water year or water doi. And again, we see that there's not any sort of hump in our data or a U in our data, which means that the model, again, is doing what it's supposed to do. So we should be pretty confident that our model is fitting pretty well. Unfortunately, the default plot for a GAM is not really appropriate for publication, but let's go ahead and walk through the useful information it gives us. First, these ticks along the bottom are what's called RUG. It's called a RUG plot, and it just shows a vertical line wherever we have an observation. Our y-axis is a little different than the input data. First of all, what it says here is it's just telling us what is this a function of. It's the water day of year, and it has the effective degrees of freedom of 8.14. But the other thing to note is that this is centered around the intercept. So the 0 line here does not mean a 0 CPV. That's actually the mean of our response variable, or the intercept. Now, this isn't suitable for publication, so let's walk through how I take our model and plot a more suitable visualization. This is a framework that I used, and you can use it for any kind of model, linear regression, logistic regression, whatever you want. The first thing you want to do is determine which is your primary predictor or predictors you want to visualize. You then choose what value to hold all other variables constant at. In our case, we only have one predictor variable, so we don't need to worry about this step. You then choose the range across which you want to simulate that primary predictor. You can use the entire range, so from max to min, but in publications, I often constrain to the 5th to the 95th percentile. And I do that because that discourages people from really paying attention to the edge of the regression where that uncertainty is high. Then we're going to use the predict function to, you guessed it, predict across this primary predictor. And we're going to use the new data argument in the predict function. As you get better at this and become more familiar, I encourage you to investigate the EM means package and the effects package. The first thing we're going to do is identify that range. So we're going to take our predictor variable, we're going to remove any and A's, and we're just going to get the range. Mod doi range is going to have two values, the maximum doi and the minimum doi. We then are going to create a new data frame called new.1, where it has one column, and that column has the exact same name as our predictor variable. All of the columns in this data frame have to have the same names as your predictors in your model, or the model won't work. And we are going to create a sequence from the minimum to the maximum doi value by an increment of 1. Or in other words, we are going to ask for a prediction every single day from the earliest day in the water year to the latest day in the water year. We then take this data frame and we plug it into our predict function where we say we want to predict our model, and we want to use this new data frame called new.1, and we want the model to show us not only the mean model prediction or the fit, but we also want to get the uncertainty around that fit. So we can use the str function, which asks R to give us the structure, and it is a list that consists of two elements within it, or our prediction and the uncertainty around that prediction. Now I encourage you to look at the handout. I show you how to take the standard error and convert that into a confidence interval, and I show you how to make the next plot I'm going to reveal. This is a much more suitable plot that we would include in a publication. We have log E transform catch per unit effort across our day of year, and we also included back transform CPU data and the actual dates. And what we have here is the line represents our model prediction, and the gray polygon is the uncertainty around that prediction. But at the end of the day, you usually run stats not to get a pretty line, but to determine something about significant differences or lack thereof. In the handout, I show two different ways to go about this, but I'm just going to show you the simplest one now. So the first thing you do is you determine what is the confidence level you want. And that is a confidence interval around our model prediction. You then take two values along the x-axis, and those are the values you want to compare. We're going to test whether our catch per unit effort at December 1st differs significantly from our catch per unit effort on June 1st. So what we do is we take the upper and lower limit at December 1st, and we compare it to the upper and lower limit at June 1st. If these uncertainties overlap, we would say there is no significant difference between the catch per unit effort at these two dates. But in our case, there is a gap, and so that tells us catch on June 1st was significantly greater than catch on December 1st. So that's a crash course in general additive modeling. Again, please check out the handout for a much deeper dive into, first of all, the under the hood section of GAMS. I go into a lot more detail and show you how to control your output. I also show you how to troubleshoot when you get some errors. But I also walk through the example of having one smoother with two different terms in it. A term for when the YOLO bypass is inundated and a term for when YOLO bypass is not inundated. And you can then compare whether or not your catch unit effort differs between inundation and uninundation periods. I also show you the other way you can have statistical inference using GAMS. And I go into how to analyze time series data, which a lot of what we do is time series data and cyclical data. And I also have a number of references that you can cite and give you additional information, including what is that uncertainty you should use around your prediction. With that, thank you for your time, and I'll happily take any questions you may have. All right, well, thank you, everyone. Hopefully you at least learned a little bit. We have about 4 minutes for questions. Feel free to ask a question about GAMS, or if you want to ask a question about any of the previous talks, if no one has any GAMS specific questions, we could field 1 or 2 of those now. While we are waiting, I'm going to try to pull up, should have thought to do this earlier. I'll go ahead and pull up the handout, try to, and show you what that final plot looks like with the inundated versus uninundated data. Get a screen share going. So in case anybody was or is curious, here's what that GAM model looks like when you're comparing the inundated to the uninundated. If I remember correctly, I also walk through the 2 different ways to derive statistical inference. And I actually talk about time series data and show you what this analysis looks like when you are trying to account for the fact that your data are actually a time series, so they're not truly independent data. With that, we have a comment that just says, nice presentations all. Thank you very much. I'm not seeing anything else. So unless there's any other questions, I'll stick around until noon. But I encourage you all to join us again at 1 p.m. Get some fuel for your brain and stick around because I, for one, am excited to see what Sam has to say about Bayesian models. Thanks for your time, everybody, and I will see you at 1. Here is a question from Jason Pasrick. I would encourage Jason for future questions for you to throw those in the Q&A. My limited understanding from GAMS is that while they are good at describing data, they are limited at predicting. What are your thoughts? I pretty strongly disagree. GAMS can be very, very strong at predicting. It just determines on what you mean by predicting. If you mean forecasting, as in predicting future conditions, then a simple GAM probably won't do it. You do need to take into account the fact that your data are time series, and the truth is you probably want to use more of a model that propagates your model uncertainty, which Bayesian framework is going to be much stronger for that. The main limit of GAMS, as I mentioned in the talk, is that you don't get those model coefficients. So I can't write a report or build a publication where I say, hey, your beta naught is 12 and your slope or beta 1 is 0.11. You can't derive that output. And so anybody that wants to replicate your model has to have your code and your raw data. Future note, panelists cannot enter questions in the Q&A. So if you're a panelist, go ahead and raise your hand. Thanks for that note, Christine. All right, everyone. I hope you learned at least a thing or two and are at least a little less scared or a little more excited about building some models in R. And I will see you all at 1 p.m.